afternoon and good evening wherever you are on the globe today. My name is Joe and I am your webinar host. Welcome to the Advancing PFAS Analysis webinar with Chiron by Zeptometrics. Thank you for joining us, whether it's here live or on demand sometime in the future. We're very pleased that you can be it. Before I introduce our fabulous presenter, I'd like to highlight a few things about our webinar platform. To ask any questions, please use the Ask a Question box, and we do encourage you to all ask questions whenever you feel the need to do that. All questions need to be typed in, they're all anonymous, and we'll make some time at the end of the presentation to read out the questions and allow Huling to answer them. If you are watching this on demand, you can still type in questions, which is brilliant. They will be emailed directly to our presenter, Huling, today, and she will get back to you directly on email. But please do ask questions. The On24 webinar platform is designed to be flexible, meaning that you can rearrange your screen to suit your viewing needs at any time during the presentation. And finally, at the end of this webinar, an email will automatically be sent to you all. Approximately, it is about an hour after the event ends, with a link to the on-demand version of this event. That means that you can share this with all your colleagues that couldn't make it today. Thank you. Now it's time to introduce our presenter. This is Huling Liu. She works as a Senior Director of R&D Analytical Reference Materials at Chiron. And she leads the R&D team in developing and producing high-quality certified materials that are absolutely crucial for accurate analytical measurements. So now I'm going to hand you over to the capable hands of Huling. Thank you.
Yes, hello and welcome everyone. Thank you for joining this webinar. So before I going to talk about advancing PFAS analysis, I will give some say some words about the company for those of you who may not be familiar with our company. So Chiron is a ISO accredited producer or supplier for reference materials. It was founded in 1983, so we have 40 years history. Chiron specializes in synthesis, isolation, and purification of analytical reference materials for environmental analysis, for food, for safety, petroleum chemistry, and forensic and toxicology. So we adapt quickly to change in the market needs and solve the most challenging problems in analytical chemistry, I would say. Since 2023, Chiron has become part of the American organization Antalya Scientific under the Zeptometrics division. So Zeptometrics offers a comprehensive range of certified reference materials and proficiency testing solutions through its 4 brands now. It's SPEX, NSI, Chiron, and HPS. So these 4 brands deliver high quality products for a variety of applications. So we offer certified reference materials in organic analysis, inorganic analysis, microbiology, proficiency testing or PT Express, and also custom development. So PFAS has been one of our strategic product lines within the environmental and food safety sectors. We have been actively developing both legacy and emerging PFAS reference material for over a decade. So today I'm excited to present the work we have been doing on the development of PFAS reference materials, especially on the certified reference materials. So I will begin with a brief background introduction. So PFAS are a well-known class of industrial compounds characterized by their variable chemical compositions. But in general, PFAS compounds share 2 common structure features, a hydrophobic tail and a hydrophilic head. The hydrophobic tail is the pure or polyfluoroalkyl alkyl chain, and the hydrophilic head is primarily the acid function group. Either is carboxylic acid or phosphoric acid and or sulfonic acid. Those at least for the common feature for the legacy PFAS at least. But for Some of the new generation PFAS compound, they are probably more derivatives on the head part, so more function groups attached to the head part. The specific combination of like the carbon chain length, type of function groups, and number of fluorine atoms in the chain gives rise to a vast array of PFAS molecules with very diverse applications. Ever since the development of these chemicals, the large group of synthetic compounds with their unique properties like waterproof, oil repel, have been effectively used as surfactants and are highly desirable for a wide range of industrial or consumer applications. So common use include the products like carpet, clothes, food packaging. The well-known commercial items like Scotch Guard from 3M, Teflon from DuPont, and the firefighting foams used by the US Navy, etc., etc. PFAS were developed since 1940s or 1950s. The products are commonly produced by two manufacturing processes, electrochemical fluorination and fluorotelomerization. These two processes, two techniques, will give PFAS products with a little bit like different structure or composition. With electrochemical fluorination, the PFAS generally have even or odd numbers of perfluoroalkyl chain. They are mixtures of linear or branched isomers with linear isomer maybe 70 to 80%, branched 20 to 30%. The carbon chain length can be variable, but the legacy one, the C8 perfluoroalkyl chain PFAS, like the sulfonate PFOS, carboxylate PFOA, and also the sulfonamide, the legacy PFAS were produced by this process. But after the phase out, the solder chain replacement, the C4 and C6 versions are also produced. Some of them are also produced in this process. Fluorotelomerization is a new or common used method nowadays. They will give even number perfluoroalkyl, mainly linear chains. It's not 100% linear chain, but mainly. And many polyfluorinated PFAS precursors like the fluorotelomere group, alcohol, sulfonamic acid, or carboxylic acid are produced by this process. PFAS compound pose significant challenges to both the environment and the human health. These so-called forever compounds are more than 10,000 substances. Someone says more than 12,000 substances across a large variety of structure classes. Most of PFAS compounds are bioaccumulated, persistent, which give the significant environmental and human health concern. The analysis and monitoring PFAS present significant technical and scientific challenges due to their chemical diversity, low environmental concentrations, and persistence. One of the major challenges for PFAS analysis is the need for highly categorized reference materials, both RMs and CRMs, to provide accurate, process, and reproducible data. So there are many factors to consider when conducting PFAS analysis, but several key elements or basic requirements for reliable analysis are outlined below. The first one is the robust instrumentation and validated methods. So it's crucial to have the instrument with high sensitivity and selectivity for PFAS detection. The second is the use of reference materials or reference standards. Reference materials need to be developed to meet evolving regulations, standardized testing method, and emerging research need. This is a constantly evolving area. We also need to understand the PFAS forms. It is important to distinguish between technical mixtures, isomeric mixtures, and pure isomers, either is linear or branched forms. This too is also ongoing changing area. So we need to understand where the analysis will be or where the regulation heading next. So we need to follow the established method and the guidelines in the regulatory compliance. Regarding PFAS regulation, the regulatory landscape is dynamic. It is a number of PFAS compounds and the regulation globally is expanding as the scientific understanding and public awareness grow. A brief summary globally for the global regulatory landscape of PFAS as for the 2025, the regulation of PFAS varies across different areas, reflecting the complexity of and evolving nature of PFAS management globally. But the sum listed here is a brief summary. The first one is Stockholm Convention on POPs, persistent organic pollutants. This international treaty listed certain PFAS compounds for regulation or elimination the use. This includes the the C8 PFOS and C8 PFOA, the well-known ones, and also the C6 sulfonic acid, including their derivatives, salts, form of them, and related compound. The EU initiative, there are two regulations that need to be mentioned. The first one is the REACH regulation, which the restriction covering more than 10,000 PFAS compound. The second one is a POPs regulation as well, because EU is also implemented this regulation in Europe. Well, in the United States, EPA has established the health advisories for certain PFAS in drinking water, wastewater, soil, air, and so on. Environmental regulation of PFAS has increased, as said, but introduction of national and international regulations means that many laboratories now have a regulatory requirement to identify and quantify the ever-evolving core set of PFAS compounds to monitor their presence in a wide array of matrices. On the left, there is some list of some examples of regulatory frameworks for different matrices, for air, drinking water, non-potable wastewater, wastewater, and industry effluents, and so on and so on. But all of these regulations of PFAS overlaps, even in different countries, different areas. The substances covered by these regulations overlapped. So altogether, there are 61 core PFAS compounds presently under regulation. The examples of well-regulated core PFAS compound is listed on the right. Well, what about those, everything else about those not under covered by the regulatory monitoring regulations? There are thousands of other PFAS compounds not currently covered by the regulation. So this can be the emerging PFAS, can be PFAS precursors, can be the intermediate degradants or metabolites. How do we start to study this compound and their impact without reliable reference standards and analytical methods? This issue, the need for high quality RMs or CRMs, this issue was highlighted in the National Science and Technology Council report for the White House published in the US in 2023. Even the Trump administration, about a couple of weeks ago, the Trump administration said that it will pursue regulation for certain manufacturers of chemicals, plastics, and synthetic fibers after pausing implementation of the similar Biden-era proposal. So it is important, crucial to have PFAS reference materials and certified reference materials. So RMs and CRM for PFAS test or analysis are crucial for quantification, identification, and toxicity testing, as alluded in the figure below. There is a new publication addressing this significance. RMs and CRMs also essential for developing and validating analytical methods, ensuring reliable data for decision making, and understanding the extent of PFAS contamination. The highly characterized reference materials need to be developed, as said, by regulator to in line with the regulatory requirement, method development, and research. So before I talk about more in detail about RM, CRM for PFAS, I want to say some words about the difference between RMs and CRM. Probably is not very clear for everyone. So by definition, RM, a reference material, is a material sufficiently homogeneous and stable with respect to one or more specified property value, so which has been established to be for its intended use in measurement process, so fit for purpose. On the other hand, a certified reference material, CRM, it is RM still, but it's characterized by a metrological valid procedure for one or more specified properties. So need to be combined with a reference material certificate that provides the value of the specific property. It should be associated with a certainty and a statement of metrological traceability. So ANAP, the National or International Accreditation Board in the US, has listed this attribute to distinguish RM and the CRM. The key factor or key requirement for CRM is one is the certified value, the second is the metrological traceability, and you need to provide a reference material certificate for CRMs. For RM, it's called product information sheet. There's another list to say the difference between RMs and CRMs. I'm not going to read more in the detail, but in a simple term to say from application point of view, RMs are more for system suitability standards. They are mostly used for method development, for research, for internal training, for screening, for example, or spiking experiment. CRMs are more to use for established metrological traceability measurement, such as for the final calibration or validation for your method, for interlaboratory studies, and for reporting to regulatory bodies. But there are challenges, both for RMs and CRMs. The first challenge is limited availability. We're talking about 10,000, 12,000 PFAS compounds, but the reference materials, especially CRMs, are not all available for them. The second challenge is the variability of quality of these reference materials. PFAS are compounds with unique chemistry and with diversity. But the regulatory bodies in many countries require monitoring of a specific PFAS compound. And the most common method for environmental toxicology PFAS analysis is LC-MS method technique, as everyone knows probably. With this targeted MS analysis using the optimized multiple reaction monitoring MRM transition with MS detection. I'm not going to talk about the technical so much, but on the left side you see the typical chronogram for PFAS standard mix. So from this program, because it is target analysis, you see what you want to see or what you need to see. But what lies behind each peak in this standard mixture What about the linear or branch isomers? Are they both present, or is it unrelated homologs in the mixture? Are the residues you don't see on the chromogram present? Are PFAS not covered by regulars and you don't specifically target it? Are they present as well? Are PFAS precursors? All this will need to give you impact on the analysis of the accuracy. So reference standards are typically produced by companies like Chiron that we either source PFAS from manufacturers or we synthesize or characterize them to meet defined purity or traceability as requirement. But some commercial available CRMs are less pure or lack the transparency regarding their purity verification. Such details are not often found from the certificate of analysis. I give an example with this PFAS CRMs. On top is this PFAS CRM we produced, developed in-house at Chiron. This is in-house synthesized. On the bottom, you'll see the chromogram for a commercial PFAS CRM. As you can see, the purity are different. So the impurities could be isomers or homologs. I need to address that it's might be okay. It's not pure as, but it need to be important to be to know that you need to verify the impurities, either it's isomer or homologs, because if you don't do you don't determine the isomer profile, the impurities, for example, they will give a negative impact for quantification. So they will contribute to system bias of unknown proportions of accuracy. And for toxicity testing, different isomers may possess different toxicity profile. So now I'm going to talk about our process for characterizing PFAS reference material and certified reference materials. So Chiron's approach is we start with synthesis. We do take technical product as well. So either synthesis or technical product, we'll first do is purification. So to remove any impurities. So after purification, we will do structure confirmation by different techniques. And after confirmation, the structure, the identity, we will do check the chromatographic purity, chromatic analysis. And then we do residue analysis to check the net purity before we certify to be RM or CRM. So what we synthesize are the novel emerging PFASs that you don't find standard from other places properly. We also do synthesize on isotope labeled, carbon-13 labeled or deuterium labeled as internal standards. We do purification to improve the purity, as said. So what we tested at Chiron first for neat material, we test the technical mixture, we test isomer mixtures or isomer purities to verify the purity or the composition. We test the single component solutions or multi-component solutions. We choose the best solvent to have the best solubility, and we want to avoid the compound interactions when you have a mixture, with several components in it. For CRMs, in the end, we need to test the stability and homogeneity, and we give the traceability on the certificate of analysis. This is the synthesis example. I will not talk about details because synthesis of PFAS is complex. Due to the complexity of the PFAS compound, we're not talk about one class of product. We're talking about a diversity of a class of compounds. But the synthesis I want to do, the purpose is to improve the purity, to increase availability. So what is listed here is a synthesis rules as an example for the sulfonamide derivatives. What I want to say is that we choose the synthesis rules, we optimize it, we choose the key intermediate to develop by optimize each step to purify each intermediate. We can manage to to get the pure final product as pure as possible at the end. This figure you have seen in the start, this is the PFAS compound we are recently working on. So we have developed the reference materials and certified reference material for this compound. I will take this as example. Actually, this result has been published in several publications. This is one of it. If you're interested, you can read them. So take the first group A as an example. I just want to show what the structures, what kind of CRMs we have developed. This is a sulfonamide with methyl ethyl propyl chain as both native and deuterium labeled internal standard. From that, we can also put another chain on the glycine chain on. So we have a second group of sulfonamide derivative with glycine. And then we can move on to the third group. We do the reduction. Then we got to the ethanol group, native and deuterated. And then we have from the alcohol, we can make the acrylate and methacrylate as esters. So those are this within this group as well. Just for you give you some idea what kind of synthesis we're doing, what kind of reference material or certified reference material we are doing. So after the synthesis, after the purification, we will need to go to do the characterization, to the analytical approach to our PFAS reference material before we certify them. We have different analytical approach for our reference materials and the certified reference material, but generally we first do the identity or composition confirmation. Depending on the type of PFAS compound, there is the structure differences. You need to choose different analytical technique. Either is GCMS, GC-feed, or LCMS, or NMR to identify the structure. NMR is mostly fluorine are mostly used for PFAS compound. And second, you need to confirm the purity composition. You can use GC-FAD MS. FAD give you a linear response. You can also use LC-UV or MS. For UV, if the compound has a chromophore, then you can do UV. Otherwise, you need to choose MS. Mass balance, I will talk about it in a minute. This is needed to check the net purity. TGA and the QNMR is the technique we use. And finally, we need to check the stability and homogeneity and so on. Now I would like to give you a brief example of the characterization grades of reference materials. In this case, I take the C8 sulfonamide as the example. This compound do not have the chromophore, so in this case, it is LC-MS, the technique used for the analysis. I will show you three different grades of product: a technical grade, crude synthetic product, and final, the purified product. On the right, you see the conditions for the analysis for LCMS. For screening, we use the SNT expert PFAS column. For final, the purity for certification, we use the standard C8 column for characterization of the purity. Yeah, as you can see on the top is a technical product. It is apparently is a mixture of the anisomer, linear isomer product, together with the branch isomer and homologs and other organic impurities. On the bottom is in-house synthesized product, but before purification, you see it is a mixture of anisomer, homologs, and there are some homologs present as well. But after purification, we managed to get the anisomer as 99.0% pure. There are tiny percent of homologs and branch isomers. This most important. You need to verify. You need to characterize the impurity. There is homolog and branch. You need to give the verification. Another important feature for RM or CRM characterization is the determination of residues, solvent, water, and inorganic residues in our product. Why that is important? I take this example. This figure shows three batches of the same PFAS product. They are same product. No corrections for residue in the RM solution. So all of the three batches have 98 to 99% chromatographic purity. So all three should be the same concentration, should have the same concentration. But as you can see, one batch, the field peak has water and inorganic residue present that gives lower detector response. So the concentration actually is not accurate if you don't correct the residue contaminant. How we do the residue analysis to give the mass balance or net purity. The first technique we use is called TGA, thermogrammetric analysis. Now, TGA is an analytical technique used for measure mass changes in the material as a function of temperature or time. The key rule for PFAS purity assessment with TGA first is the detection of volatile impurities. Volatile impurities like water, solvent, at temperature increase, the mass will change because the volatile contaminants disappear. So this is one of the technique, the area we use TGA. The second is for the quantification of non-volatile residue like the salt or ash, because they can remain as non-decomposable residue as a temperature at high temperature. So in the end, we can calculate how much the residue inorganic ash in the sample. The data shown here indicate low water or solvent residue content for this sample we have analyzed by TGA. But we need to note that to our knowledge, so far, TGA method worked well for PFAS analysis with the longer chain PFAS. The chain length is bigger than C11 carbons. Although shorter chain PFAS, the method does not work so well because shorter chain PFAS have low melting point. They tend to be sublimated when the temperature increases. Then we will have to use another technique, which is QNMR. QNMR is very powerful technique because it directly quantify the amount of the analyzed by using an internal standard. So by comparing the internal standard signal, in this case a CF3 in this internal standard, this is the signal, integrate the area, compare the same signal from the same function group in this analyte. You compare to the integration area and you calculate the purity of the analyte. It's a very powerful technique, but it's really required the sample is pure and have a good resolution on NMR. It's also need a lot more materials to run the NMR, as people may know. So there's some advantage and some limitations with the technique, which I will not go in very much in detail. So to summarize the development of PFAS RMs and CRMs, we have developed the synthetic method for do the PFAS compound synthesis purification in order to get purer isomers, purer products, to increase the availability and purity. High purity product is our purpose. We characterize our PFAS reinforced material with different analytical techniques for chromatographic analysis, like the LC, GC, NMR, to 12, depending on the complexity of the sample. We also check the neck purity to check the residue, which is important to give the accurate concentration. So as ISO 17034 accredited reference material producer, we actually have more than 400 PFAS compounds in our stock, including the legacy or emerging PFAS, precursors, intermediate, degradant products, and so on. But recent years, or in the last 1 to 2 years, we have been focusing on the PFAS CRMs development. So far, we have developed 40 PFAS CRMs. We have characterized them, validated them to ensure high purity and accuracy. So the CRM will contribute significantly to the accurate and reliable monitoring of PFAS environmental and biological samples. If you are interested in more in detail which PFAS CRM we have developed, please visit our website, or with this, we have a brochure with a QR code, and probably you can see more in detail. On this one, I just want to show you the certificate as example. On the left hand is the for the RM. For RM, the document is called product information sheet. Is not a code certificate, but for CRM, it's a reference material certificate. So each has different documentation and different content because a different requirement for certification for RMs and CRMs. This is just continue for the first page. I will not talk about more. For future, we will continue the development of new emerging PFAS, either RMs and CRMs. We will continue to do the development of CRMs. Now we're focusing on the mixed PFAS mixes CRMs for the regulatory compliance. We are continuously working on the synthesis of carbon 13 labeled PFAS internal standards. On the right is the list of PFAS internal certainty labeled PFAS reference materials which are available right now. So those are the fluoropolymer type of compound under the acid. We will work more and more new compounds will be coming soon to our website or to the market. So with that, we will acknowledge the funding we have been receiving from EU Commission, from the Regional Research Council, and so on, for help us with R&D and for QA, QC, for the development of certified reference materials for PFAS. And once again, I will thank you for joining us for today for this webinar. So please feel free to submit any questions in the chat, or just let's know. Let's start with discussion, and I'm happy to answer any questions you may have. Thank you once again.